Hi, and welcome to my OCR A-level biology revision session with me, Christine. So in this lesson, I'm going to look at meiosis. Now, before I talk about meiosis, I want to take you back to mitosis. So if you haven't already checked out my video on mitosis, then please do check that one out. But just as a quick recap, mitosis we know is important for growth, for tissue repair and asexual reproduction. And mitosis we know has 4 phases to it: prophase, metaphase, anaphase, telophase. And at the end of mitosis, we end up with 2 genetically identical daughter cells. Well, what's important for us to note is meiosis is actually necessary for the production of gametes. And the reason it needs to produce gametes is that gametes are necessary for sexual reproduction. So we actually call meiosis a reduction division. The reason it's a reduction division is because it actually is made up of 2 stages: prophase, metaphase, anaphase, telophase 1, and prophase, metaphase, anaphase, telophase 2. So it's a reduction division because we're going to take our parental cell, our original cell, and we are going to result in having 4 cells which are half the genetic information of the original cell. So we say we've gone from a diploid cell, 2 sets of every chromosome, to a haploid cell which has 1 set of every chromosome. So we're going to look at how that happens. Now, if we look at prophase 1 then, and we know that in prophase, our nuclear envelope and nucleolus disappears. We know that our chromosomes have condensed, they've supercoiled. We know that our centrioles have moved to the poles and they are producing the spindle fibers, which are part of our cytoskeleton, and they will attach at the centromere. So we know that DNA replication has occurred, and that has resulted in us having these sister chromatids which are attached at the centromere. So what we need to note then is, because we're in meiosis, in prophase 1, the key distinguishing difference is that our homologous chromosomes are going to pair up. Now, homologous chromosomes are chromosomes that are the same length, with the same genes at the same loci. So you can see quite clearly that I have 2 different sized chromosomes here. I have a set of homologous chromosomes which are short, and a set of homologous chromosomes which are long. So they are homologous chromosomes because they are the same length, they have the same genes at the same loci. Now, it's important you note that there are different alleles that can be present on those chromosomes. So a gene is a section of the DNA molecule, whereas an allele is a different version. And I've represented this with my blue allele, for example, and my red allele, but they're in the same position because it's the same gene. It could potentially be petal color, for example. So if we understand that during prophase 1, our homologous chromosomes are going to pair up as a pair, which we call a bivalent, the next thing we have to know is that during prophase 1, if they are long enough and if they intertwine in such a way, they will have something happen to them which is called crossing over. So crossing over is where they are intertwined and they can exchange the genetic material at the point in which they have crossed over. Now, this can only happen at prophase 1, and the crossing over occurs between 2 non-sister chromatids. So it's important to note that my homologous chromosomes, there is one that is pink and one that is purple. So the pink one we'll say is the maternal chromosome, and the purple ones are the paternal chromosomes. So the only way in which crossing over can occur is if one maternal chromatid and one paternal chromatid have crossed over, and that will result in an exchange of the genetic makeup. So that's important for us to note, in a way in which we can result in variation, which gives us the genetically different daughter cells. Now, if we remember in metaphase, then we know in metaphase of mitosis that the chromosomes line up at the equator at that metaphase plate. Well, it's exactly the same with regards to meiosis. During metaphase 1, This time, however, it's the homologous chromosomes that line up at the equator. So the homologous chromosomes are going to line up at the equator together. Now, it's important that we note, how do they line up? So if we think about the pole which is to the left-hand side of the screen and the pole which is to the right-hand side of the screen, you can quite clearly see that I have a short paternal and a long maternal, which will move towards the left side of the screen. And we have the short maternal and the long paternal, which will move towards the right side of the screen if this is representing my cell and the chromosomes being pulled to the equator. So if they line up like that, that is how they will be separated. Well, actually, it is independent of each other in how they orientate themselves. And we say that is called independent assortment. So have they lined up paternal with maternal, or have they lined up paternal, paternal, maternal, maternal? So however they line up at that metaphase plate during metaphase 1 is the first part of independent assortment. And what will then happen is during anaphase 1, our homologous chromosomes are going to be separated as those spindle fibers retract back to the poles. So as these proteins are brought back into the centrioles, they are going to pull those homologous chromosomes apart. As they're being pulled apart, that's when the exchange of the material actually happens at the chiasma, which is the point at where the crossing over has occurred. So you can quite clearly see in my diagram here that we have had an exchange in the bottom section of our chromatids, where there is a part of the paternal which is now moving to the left side of the screen and a part of the maternal which is moving to the right side of the screen. So after anaphase 1, we then have telophase 1, where they reach the poles, where the nucleus envelopes reforms and they uncoil, and then we have prophase 2 occur. In prophase 2, they then disappear that nucleus envelope. They then will supercoil again, and we will have those spindle fibers reforming. Now this time what's going to happen is we have now had two cells which are haploid because they contain half the genetic information. They have one set of every chromosome. So instead of having two, they now only have one. So that was that first reduction division. We've gone from a diploid cell to haploid cells. Then metaphase 2, they are lining up at the equator again. So this time, because there's only one, that's the only ones which can line up at the equator. And what's going to happen then is after they have lined up at that metaphase plate, during anaphase 2, our sister chromatids are now pulled to the poles. So it's important to note that during the anaphase 1, our homologous chromosomes were separated. Now during anaphase 2, it's our sister chromatids that are being pulled to the poles. So once they have been pulled to the poles, then we get to our telophase 2, and our chromatids have reached the poles. They're now chromosomes. Our nucleus has reformed, our nucleolus has reformed, and cytokinesis can occur. So remember, our PMAT1, PMAT2 occurs. The key difference is whether it's the homologous chromosomes that are being lined up and then separated during the PMAT1, and during the PMAT2, it is the chromosomes and therefore the sister chromatids that are pulled apart. So that's the reason that we call this a reduction division. So meiosis is a reduction division because what we're actually producing is 4 haploid cells. So 4 haploid daughter cells that are genetically different from the parent. So they are not the same, and they are not the same if they have crossing over occur. So independent assortment and crossing over are the two ways in which through meiosis variation can occur, because we are doing a reduction division. We are going from a diploid cell, which is 2N, to a haploid cell, which is N, and that refers to the number of each of the chromosomes. So if you like this video, then please do click on the like button and subscribe to my channel. Also, if you haven't already done so, please do check out my revision platform, www.aiqchat.com, to help you to revise.